Hey guys, welcome to our review of the cell membrane. I want to first start by talking about this diagram of the cell membrane here at the top of the page. Now, the cell membrane is a really important part of every single living cell. It forms what we call a semi-permeable barrier around the cell. That means that it allows certain things to come in and out of the cell, but blocks other things from coming in and out. Some people refer to the cell membrane as the fluid mosaic model, and here's why. Fluid refers to the part about how different parts of the cell membrane can move around. They're not stuck in one place. And mosaic, kind of like you've heard about in world history class of mosaic pictures, mosaic means made of small pieces, and you can see there are many small pieces in this diagram of the cell membrane. Those pieces include the phospholipids. This is a enlarged version of a phospholipid, but it shows the head and the tail. Now, in our phospholipids, we're going to actually form not just one layer of phospholipids, but two, and for that reason, we'll call it a phospholipid bilayer. Now, within that bilayer, you're going to notice a certain arrangement. The heads, which are these kind of blue circles, they are on the very outside of the cell and then on the inside of the cell membrane. Those heads are what we would call polar, and they're hydrophilic. That means they love water and they're okay with touching water that might be outside of the cell out here or inside of the cell here. Those other parts of the phospholipid would be the tails, and those are made of fatty acids, and those tails are nonpolar. They're hydrophobic. What that means is they hate water. So they're going to stay in the middle of our cell membrane away from where water might be. The next important part of our cell membrane is going to be our cholesterol. And if you look at the cholesterol there, that is a specific part of the cell membrane that is going to improve the stability. So if it starts getting too warm and those phospholipids try to move away from each other, the cholesterol is going to hold them in place. If it's too cold and the phospholipids try to cram together, the cholesterol is going to keep some space in between them. It's going to improve the stability of the cell membrane overall. Now, the next and probably most important part of our cell membrane is going to be our proteins. Our proteins are set up in different ways. There are many different types, which we're going to talk about, but the main function of those proteins is going to be to allow materials to move in and out of the cell membrane as needed. They are for transporting materials in and out of the cell. Now, we have a couple different types of proteins. The first that we will talk about are these that go all the way through. You can see some examples there. If they go all the way through, they're called integral proteins. If they're just on the edge, like this one, they're called peripheral proteins. Just like your peripheral vision is just out of the side of your eye. These are just on one side of the membrane. And then up here, you can see another peripheral protein, but that one's special because it has a carbohydrate chain up here. And if it has a carbohydrate chain attached to the protein, it's called a glycoprotein. And that glycoprotein is going to be used to receive signals from outside of the cell. So this cell membrane is a very important part of the cell. It goes actually all the way around the cell. So it's not just a little line, but it would actually go all the way around a circle of a cell. And it allows certain materials to move in and out. Now, with cell membrane, we also talked about how materials move across the cell membrane. And if you look at this blue box over here, what you're going to see is a description of passive transport. We had a little jingle in class. We said if it's going high to low, it's going with the flow, it's passive transport. And what that means is if materials are moving from an area of high concentration to an area of low concentration, they're going with the concentration gradients. They do not require any energy, and so that would be passive transport. So high to low, go with the flow, it's passive transport. That includes diffusion of certain materials, like, for example, if you put food coloring in a glass of water, it will move the particles from where it's highly concentrated to spread out to where it's low concentration. That's passive transport, no energy needed. There's also osmosis, which is a special type of diffusion. It's the diffusion of water. So if you hear osmosis, you just need to know that water is moving across the cell membrane. And then there's also facilitated diffusion, and that happens to be when we go from high to low, going with the flow, using a protein, but still not using any energy. So that's all passive transport. Next, we talked about active transport, and active transport is shown down in this orange box down here, and it's kind of different from our passive transport. So our little jingle for that was, if it goes low to high, it goes against the tide, it's active transport. So low concentration to where it's high concentration goes against the concentration gradient. We call that active transport. And if you are going against the concentration gradient, just like, let's say if you were downstairs at the end of a school day and it's 2:55 and you decide, oh gosh, I forgot something upstairs, you're going to go against the concentration gradient. And most people are moving downstairs, you're going to be moving upstairs. That's going to take more energy. So low to high, you're going against the tide. That's active transport, needing energy. We talked about several different ways that happens. There was active molecular transport, which uses an integral protein but also uses ATP energy, which was made during cellular respiration. We've also talked about endocytosis, which is when materials move in large chunks. Actually, it requires the cell membrane to kind of wrap around those materials and bring them in. The phagocytosis is when the cell eats kind of larger particles. So cell eating is phagocytosis, and cell drinking, when it's just taking liquid in, is called penocytosis. And then we had exocytosis, which is when there's some material inside of the cell and it needs to be pushed out. The cell membrane has a way to push it out. And active molecular transport, which uses ATP and the integral protein, endocytosis and exocytosis are all examples of active transport. All right, for our next idea here of cell membrane transport, let's talk about osmosis. These are the beakers we use to talk about osmosis. And we want to talk about osmosis as an example of passive transport. Osmosis is an example of when there is movement of water from where there's a lot of water to where there's not a lot of water. It's high to low, going with the flow. It's passive transport, but osmosis is specifically that diffusion of water. And when we're talking about these osmosis beaker problems, we usually actually don't look at the concentration of water. We focus more on the concentration of the solute. Now, if you remember, a solute could be something like salt or sugar that is dissolved. And what we look at, and have a little jingle to remember that, is that water moves to hypertonic areas where the water concentration is actually lower, but the solute concentration is higher. So osmosis, water moves to where it's hypertonic, where there is a higher concentration of solute, like sugar or salt. So we had these words, hypotonic, hypertonic, and isotonic. Hypotonic meant a low concentration of solute. Hypertonic meant a high concentration of solute. And isotonic meant that there would be the same. And when we looked at these beaker problems, we wanted to figure out where would water move within this beaker. So first we see this beaker, and it has a cell. And if we look at this cell inside of this beaker, we're going to see that that cell has 10% solute, and the liquid that it's in has 37% solute. So we can mark the cell as being hypotonic because it has a low concentration of solute. We can mark the liquid in the beaker as being hypertonic because it has a higher concentration of solute. Once we've marked those as hypotonic or hypertonic, remember hypo is where there's low solute, hyper is where there's high solute, we can determine which way water will move. Water always moves to hyper. So in this example, you can see that water is going to move out of the cell and into the beaker because the beaker was hypertonic compared to the cell. That it's going to mean that water moves out of the cell, and that cell is actually going to end up shriveling up just like our egg did in the egg experiment. That egg kind of shriveled up. So that's osmosis. Now, if we had an example where the concentrations of solute were the same, that would have been isotonic, and water would have moved in and out of the cell equally as it would have moved in and out of the beaker. If we had a different situation where the cell was hypertonic and the beaker water was hypotonic, the water would have actually moved into the cell, and that would have caused the cell to possibly swell and burst like the egg on the last day of our egg experiment, where the egg was super big and full of colorful water. But Main thing with osmosis, you want to pay attention to the concentration of solute, not the concentration of water. You want to label that. So if it's low solute, it's hypo. If it's higher solute, it's hyper. And water always moves to where it's higher solute concentration. So that's our osmosis beaker problems. Last little bit of information to know is that all cells must stay small. And the reason is that in order for them to have a good surface area to volume ratio, they've got to be small. If a cell gets too big, there's too much volume within the cell. And materials can't easily move from the middle of the cell outside of the cell, so cells stay small so that they can have a good surface area to volume ratio. In other words, cells stay small so that materials can move in and out of them more easily.